Let's address the nucleus and the ribosomes. The nucleus, it's a very complex structure made up of the nuclear envelope, right? That nuclear envelope has actually two membranes. You have the outer membrane and the inner membrane that makes it an envelope, so the nuclear envelope. That nuclear envelope has teeny tiny holes. These teeny tiny holes are nuclear pores, okay? These nuclear pores can be actually seen right here in the micrograph, right? That was obtained with an electron microscope. The nuclear pores are these areas where some material can leave the nucleus to go outside in the cytoplasm. Likewise, the material can leave the cytoplasm to come inside in the nucleus, right? The nucleolus, it's where ribosomal RNAs are produced. Okay, ribosomal RNA, so the RNA that are structural part of the ribosomes, this is where they are produced. And this is also where the assembly of this ribosomal RNA starts. However, it is not completed there. Okay, it is not completed there. The assembly of ribosomes is completed in the cytoplasm, right? Then we're going to have chromatin, and chromatin again, it's DNA mingled with some proteins. That's what we call chromatin, and that DNA is relatively diffuse, right? We have the nucleoplasm. The nucleoplasm, it's a semi-fluid substance that is the equivalent of the cytoplasm, but this time in the nucleus, right? Now, that nucleus is the one that is going to carry the genetic information that is responsible for the way the cell behaves, right? Now, going outward, we see that the nuclear envelope here expands in a series of flattened sacs. These flattened sacs have little spots attached to them. These little spots cause that structure to look rough. This is the rough endoplasmic reticulum, okay? This is the rough endoplasmic reticulum. That rough endoplasmic reticulum has an open space that we call endoplasmic reticulum lumen. That's the yellow part in here, the space right here. That's the endoplasmic reticular lumen. Now, the reason why it is important to mention this, it's because the red dots that are right here, which are ribosomes, occur in two forms. You have free floating ribosomes that are floating in the cytoplasm, right? And you do have ribosomes that are attached to the membrane of the endoplasmic reticulum. Now, what causes this ribosome to be attached to the membrane? Well, the answer is that whenever genes are being expressed in the cell, DNA is converted into RNA. All right? DNA is converted into RNA. That RNA that contains the information for a protein to exist is going to be carried from the nucleus to the cytoplasm. Okay? In the cytoplasm, this is where ribosomes are going to be located. These ribosomes are free-floating. And these ribosomes, their job is to read the information that is in the RNA to convert that information into proteins. All right? So the process by which we go from DNA to RNA is called transcription. transcription. So we are going from a DNA language to an RNA language. And from RNA language to protein, we call this translation. Translation. All right? So the ribosomes that are free-floating here are going to start producing proteins out of the messenger RNA. We call it messenger RNA because it carries a message, right? That message is responsible for the very existence of proteins. Now, some proteins are designed to stay in the cytoplasm. Other proteins are designed to be sent either in outside of the cell, or they can be integral part of the plasma membrane. You remember that the plasma membrane, it's a fluid mosaic, right? And mosaic because proteins are embedded. So you have membrane proteins. These membrane proteins are embedded in the membrane because they are produced through the ribosomes that send the protein in the lumen of the ER. So specifically, this ribosome right here will start doing translation, and at some point the system is going to notice that, well, this protein that is being synthesized doesn't belong to the cytoplasm. It belongs somewhere else. Therefore, what is going to happen is that this ribosome that was free-floating is going to come and attach to the membrane of the endoplasmic reticulum, now causing the membrane of the endoplasmic reticulum to look rough. But in the process, they are synthesizing a protein that will end up inside of the lumen of the endoplasmic reticulum, as you can see here. Okay, that's the protein. Now that that protein is there, that protein is going to follow its fate, right? Which fate can be sent out of the cell, right? It can be ending as part of the lysosome. It can be as part of the plasma membrane or any other membrane, right? Different fates, but let's close that parenthesis. So you understand that the ribosomes, their job is to do protein synthesis. Some ribosomes are free-floating. Other ribosomes are going to go and attach to the endoplasmic reticulum membrane. All right? Now, the endomembrane system. The endomembrane system is going to include the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum is going to involve protein synthesis. Why? Because ribosomes are synthesizing proteins attaching to the endoplasmic reticulum, causing that endoplasmic reticulum to look rough. In the process, the synthesized proteins is going to end up in the lumen of the endoplasmic reticulum, right? And there, the protein is going to be modified after synthesis, okay? That modification determines the fate of that protein. Then we have the smooth endoplasmic reticulum. That smooth endoplasmic reticulum that we can actually see right here, this is the smooth endoplasmic reticulum, is going to be for lipid synthesis, okay? That's one function. Another function is going to be detoxification. If you think, for instance, of other drugs that we ingest when we are sick, right? This drug don't belong to our body. Our body is actually going to discard these drugs, right? When we are sick and actively taking drugs. So the smooth endoplasmic reticulum is what is responsible for that detoxification of our body, okay? Also, for some cells, including muscle cells, the smooth endoplasmic reticulum, its job is to mainly store calcium, because calcium is essential for muscle contraction. Without calcium, forget about muscle contraction, right? That's why as we grow old in age, it is very important that we have a diet that is enriched with calcium, because with that additional calcium, our muscles are going to be much more healthier, and we can exercise and be able to keep um, a good tone in our muscles. But let me close that parenthesis. All right, going forward with the endomembrane system, we talked about the rough endoplasmic reticulum and the smooth endoplasmic reticulum. At this point, let's talk about the Golgi apparatus. The Golgi apparatus, its job, actually, first of all, this is the Golgi apparatus right here. The job of the Golgi apparatus is to modify lipids and proteins. They are going to sort the proteins and send them based on the signal that is on these proteins. Again, some proteins are designed to be part of the plasma membrane. These are membrane proteins. We have some proteins that are designed to be sent outside of the cell. For instance, when you consider antibodies, when we are fighting off an infection, right? Our white blood cells, the B white blood cells, are going to synthesize antibodies, send these antibodies outside of the cell, and these antibodies are going to end up in our blood, right? So the Golgi apparatus is responsible for modifying this protein so that they can be sent outside of the cell. But the Golgi apparatus is also that organelle that is going to cause the development of lysosomes. And the lysosomes are these sacs that contain digestive enzymes. These digestive enzymes are produced from the rough endoplasmic reticulum to the Golgi apparatus, where they are modified, where they are modified to end up as part of the digestive enzyme here. Once they end up in the lysosomes, then when we engulf particles such as bacteria or viruses, the vesicle is going to merge with the lysosome, and the bacterium or the virus is going to be digested with all these digestive enzymes. These digestive enzymes include lipases, so lipases. We have proteases. We have nucleases, okay, and many other enzymes, okay. So the Golgi apparatus is also going to secrete some chemicals outside of the cell. So we have some secretory vesicles here, okay, that are going to come and fuse with the plasma membrane and be able to release the content out that way. We have some transport vesicles here, and these transport vesicles secure the migration of chemicals from the rough endoplasmic reticulum to the smooth from the rough endoplasmic reticulum to the Golgi apparatus, and from the smooth endoplasmic reticulum to the Golgi apparatus, and so on. So these transport vesicles are very important, okay? We have the smooth ER here, synthesis of lipids and detoxification and other functions. All right. So we've talked about the nucleus, the ribosomes, and the endomembrane system. Now you have some membrane-bound structures that are not actually directly part of the membrane, the endomembrane system. Well, they are somewhat associated with the endomembrane system, but they constitute structure of their own. We have vacuoles. Vacuoles occur a lot in protists, right? Protists. Protists are single-cell eukaryotic organisms that look like, that are animal-like, right? This protist, and here we do have the case of, which one is that? A paramecium, if my memory is good. This is a paramecium. Okay, and this paramecium has vacuoles. Okay, and some of these vacuoles are contractile vacuoles. Okay, I'm not going to bug you with the details of what contractile vacuoles are. And then we have in plant cells, we do have this central vacuole that is humongous, right? That allows the cell to actually store some water and chemicals of all kind. And then we have the vesicle. The vesicles in general are smaller than vacuoles. Vesicles are smaller than vacuoles. Okay. All right. So the chloroplast in plant cells only occur in plant cells, by the way, between animals and plant. They only occur in plant cells. Chloroplasts are responsible for harvesting light energy, converting that light energy into chemical energy. And that chemical energy, ATP, actually is going to be responsible for fueling the reactions that consist of bringing carbon dioxide and water together to form sugars. Eventually, at the end of the day, you have production of glucose, which is the main sugar that is actually the byproduct of photosynthesis. So the chloroplasts, they capture light energy, convert that light energy into chemical energy, and that chemical energy is used for photosynthesis, which is the synthesis of organic molecules, right, using light energy. All right, so how about mitochondria? Mitochondria are also known as the powerhouse of the cell. They are known as the powerhouse of the cell because these organelles are the very one that actually produce much of the ATP that we use to sustain our physical activities. So I could not do all this talk I'm doing, okay, unless I had enough ATP to actually stimulate my muscles and get my muscles moving, because to talk, I'm activating my muscles and working my muscle out. Now, there are a few things I would like to attract your attention upon here. One, it's that this mitochondria, they do have two membranes. We have an outer membrane here that looks brown and an inner membrane here with the outer surface that look yellow and the inner surface that looks purple, right? In between, you are going to have, okay, well, not in between, but right here you have a matrix, right? That's the matrix of the mitochondrion, while between the outer membrane and the inner membrane, you have an interspace membrane, okay? And this foldings of the inner membrane is called crista, one crista, several cristae, okay? So mitochondria are very much responsible for the metabolic process that we call cellular respiration, which is responsible for the major amount of ATP that we found in our cells, okay? And ATP again stands for adenosine triphosphate. All right, the cytoskeleton. The cytoskeleton, it's a mix of many kind of protein fibers. These protein fibers occur in three kinds. One is the microtubules. The microtubules are going to be organized like this, like little beads that are organized in such a way that you have a structure that looks like a straw, okay? These microtubules that look like a straw are relatively stiff, right? They are very much involved in cell division. Because the structures are stiff, they allow vesicle to move along the length of this microtubules, right? They actually function like railroads, okay? On a railroad, you have train cars that can run, right? These train cars can only follow the direction of the railroad, okay? And wherever the railroad are going, this is where the train is going, right? Same thing here. Wherever the microtubule is going, this is where the transport vesicle is going. Now, the microtubules play also a very important role in cytoplasmic streaming. What we mean by cytoplasmic streaming is that the organelles in the cell occupy a certain spot at a certain time. What causes these organelles to occupy a certain spot at a certain time? It's basically how the microtubules are arranged, right? So that's what we call cytoplasmic streaming. And finally, but not least, the microtubules are also very much involved in cell division. Why? Because the microtubules are responsible for distributing DNA, the genetic information, between the two future daughter cells so that the two can inherit the same amount of genetic information. Okay? So again, microtubules are responsible for the equal distribution of the chromosomes between two daughter cells. Then we have the microfilaments. The microfilaments are these filaments that are made up of proteins, building block proteins that are called actin filaments, right? These actin proteins come together to form actin filaments, and these actin filaments are responsible for the crawling movement of, for instance, white blood cells that are running after a bacterium to eat that bacterium so that that bacterium doesn't cause us to get sick, right? So the movement in our body are caused by the microfilaments or actin filaments. And then we have the intermediate filaments. The intermediate filaments are less well characterized compared to microfilaments and microtubules, right? These intermediate filaments are also found underneath of the nuclear envelope toward the inside of the nucleus. All right, the cytoskeleton, if we continue on that, we have the centrioles. The centrioles, actually, you have two structures, okay? These two structures are starting point for the microtubules that are going to develop the mitotic spindle. This mitotic spindle that are essential for attachment on the chromosomes so that the chromosomes can be equally distributed. I spare you the details here because there are a bunch of details here, but we don't have the proper time, and this is not the proper course actually to address these details. At least if we were to address these details, we are going to address them at a later time. Okay? So the centrioles are essential for the distribution of chromosomes during cell division. I said it. The flagella and cilia, the flagella are going to be different. The flagella in eukaryotic cells are going to be different from the flagella in prokaryotic cells in a sense that the flagella in prokaryotic cells are not covered with the plasma membrane. However, the flagella in eukaryotic cells are covered with a plasma membrane. They cause movement by performing a whip-like motion. A whip-like motion, so like this, okay? That's the kind of motion that is going to cause the sperm cell to be propelled so that it can go toward the egg and fertilize the egg, right? So that that flagellum and the cilia, they have a similar structure in the sense that they are made up of microtubules and they are covered with the plasma membrane. All right, so here we do have the structure of the flagellum in eukaryotes, right? So we do have here the plasma membrane, you see, and you can see how the plasma membrane is actually covering up this structure of microtubules, right? So we have some microtubules that are making up this filament, and then you have a central part, okay? I spare you the details here. So here, this is a micrograph of a flagellum. Now, outside of the cell, we are going to have the extracellular matrix. The extracellular matrix is what allows cells to attach to surfaces, right? These surfaces cause cells that are in a certain environment to actually end up forming a tissue, okay? So here we have the phospholipid bilayer, right? To that phospholipid bilayer, you're going to have membrane proteins. These membrane proteins are attached to structures outside of the cell. These structures are complex proteins, okay? And these complex proteins are protein-protein interaction, but also protein-carbohydrate interactions, right? And I'm going to spare you the details here. So you have the cytoplasm inside of the cell. We have the cytoskeletal filaments, okay? Here we have the plasma membrane. Here we have the receptor proteins. Here you have some fibers that are called collagen fibers, where the cells are going to attach. We're going to have elastic fibers that are going to be much thinner than collagen fibers. This is also going to be point of anchorage of cells. And we are going to have polysaccharides, okay? The polysaccharides that are going to be attached to different components, including proteins. All right, so now we talked about the different parts of these cells. Now, let's ask the following question. How are cells coming together? Cells are coming together through cell junctions. There are three type of junctions I would like to address here. We have the adhesion junctions, the tight junctions, and the gap junctions. The adhesion junctions are the type of junctions that are happening between cells that do not need to be tightly packed together, okay? It is okay to have a certain space between cells. And this is what you see here. You have a certain space here. And even if you zoom here, you're going to see a certain space, right? The tight junction, on the other hand, do not leave any room for leakage, so that you are going to find the tight junctions in organs that house some liquid. For instance, the bladder, the urinary bladder. That urinary bladder has to contain urine, right? For the urine to not leak out, you need these tight junctions to develop, okay? So we have membrane of a cell 1 and membrane of a cell 2, and we do have almost like a sewing that exists between the two cells through these proteins. And then we have the gap junctions. The gap junctions are these junctions that allow two neighboring cells to directly communicate. So the information goes from cell 1 to cell 2 through this channel at no time, okay? This has the advantage that when a signal comes, a stimulus comes, and enter our, I'm just saying anything, the cardiac muscles, for instance, right? Before one side of the muscle can develop a response, the rest of the cardiac muscle has already received the signal, so that when they respond as a unit, right? And that response as a unit is called syncytium. All right, so now the plasmodesmata. I did mention the plasmodesmata. The plasmodesmata play basically a similar role as the gap junction in animal cells, but the plasmodesmata are going to occur in plant cells. And here we have micrographs of plasmodesmata. Now, what is the summary of this chapter? So as a word of summary of this chapter, we're going to have two categories of cells: prokaryotic cells and eukaryotic cells. Prokaryotic cells are very small compared to eukaryotic cells. Prokaryotic cells have a size that varies between 1 and 5 micrometer, while animal cells have a size that varies between, that is relatively larger than 50 micrometer. Actually smaller, smaller than 50 micrometer, okay? 50 micrometer, right? Most cells. Then DNA in prokaryotic cells is going to be in the nucleoid region, while in eukaryotic cells, it's going to be in the nucleus. Then we have organelles that are not present in prokaryotic cells, while they are present in eukaryotic cells. And the chromosome for bacteria, it's one single chromosome, which most of the time is circular, while in eukaryotic organism, where you have oftentimes multiple chromosomes that are linear. And then, what are the different components of a eukaryotic cell? You're going to have, among eukaryotic cells, organelles, you are going to have the plasma membrane, the nucleus, the ribosomes, the vesicles, the rough endoplasmic reticulum, smooth endoplasmic reticulum, Golgi apparatus, lysosomes, chloroplast, mitochondria, cell wall, cytoskeleton, and flagella and cilia. Now, these structures that we just mentioned are all the structures that we covered between animal cells and plant cells, right? But animal cells do not have chloroplast. Animal cells do not have a cell wall, okay? Just keep these things in mind. And please also review a description of each of the items. For instance, the plasma membrane is going to enclose the cytoplasm, regulates interaction with external environment, right? And you can do the same thing for each of the organelle. Okay, thanks for your attention. Now, in the conclusion, as we get done with this chapter, I would like to remind you to write a small review of the chapter. Go at the end of the book chapter and then write down the summary. Now make sure that the summary is similar to what we are covering in class. Do not take as part of the summary something that is beyond what we are covering in class. That's just for your information. 2, assess your understanding of the material, right? What this is going to do is that sometimes you're going to assume that you know the material until you actually test yourself. Unless you test yourself, you're not going to know if you understand the material or not. So the end of the chapter in the book contains some questions that you can do, and also you can design your own questions, and finally form small groups and reach out to each other and be supportive of each other, right? And finally, but not least, create a handwritten concept map for this chapter. Thank you for your attention. Bye now.